could talk about those instead. Oh, there we go. So, good afternoon, everyone. I'd like to start by saying thank you very, very much for having me here. I was sitting with a group outside for a little bit during lunchtime and realising maybe it's just because I live in the UK. That's the first time I've sat in the sun for quite some time. Anyway, it's very, very nice to be here. I'm going to talk a bit, and as quickly as I can manage, about the work my group does, the Oxford Protein Informatics group, looking at trying to build the computational, and it is both the databases, because that is such an important part of this, and the tools that are needed so that we can actually think about how we would design new molecules, but also understand when they'll work and when they won't. I'm not going to go through all of this slide. I'd sort of love to, but nobody ever gives me a talk long enough for me to do that. This is a very rapid collection of most of the tools that are kind of, I would say, still useful from my group. There's a lot of older ones that are not on this list, but you could play with them if you like, but I'd probably stick to things that are on here. Very quickly, if I just go from the top, you've got AIRRR-C, which numbers antibodies, HUMAB, that humanizes them or tells you if it's human, a couple of language models, which I'll talk about a little bit more, a method for rapidly searching databases of antibodies, building antibody structures, which I'll also talk a little bit more, also nanobodies and TCRs if you happen to like those, predicting antibody-antigen binding, doing the inverse folding problem that Jeff mentioned, so if you're given a kind of structure, what sequences would fit on that, profiling, so looking where can you tell where the epitopes that things will bind to, looking at the developability, can you predict whether this antibody will be good as you try and progress it to the clinic, predicting the paratope, predicting the epitope in another couple of ways, and then something about predicting affinity. Perhaps more important than all of that is the stuff on the right-hand side. So a couple of these have already been mentioned by Jeff and I think others in their talks. One of the things I realised a very long time ago was that almost everything we try and do as a computational scientist relies on the data we have. It relies on how good that data is, how clean that data is, how much we have thought about. I mean, one of the questions that just came up, how well have we checked that we're not cheating? So are the test, train and validation split in ways that mean we actually know whether this works or not? So my group has spent a lot of effort and time in building these databases. So you've heard the observed antibody sequence database mentioned already. That has both paired and unpaired antibody data in it. It's well over 2 billion now, 2.5 billion, something like that in there. SABDAB has been mentioned already. That's the collection of all of the available antibody structures. It also has all the nanobody structures in there if you happen to prefer those. Going downwards, TheraSABDAB, that's the therapeutics collected, so you can go and see everything that's got through to stage 2 clinical trials, and then go and see what that looks like. We link it to all our other databases, so you can see if we know anything else about it. More recent databases, PLABDAB, this is where we've gone and pulled out all the information from the patent literature. So this is a collection of antibodies, there's about 80,000 in there, where every single one of them has functional information associated with it. We've also predicted the structures of that and linked it to everything else, so you can use that to start looking at functional data sets if you want to. COVID antibodies, STCR-DAB, pretty self-explanatory. And then I put a green thing around the last one just to remind me to say, brand new observed TCR space. So those of you who like TCRs better than antibodies, there's about 1.5 million non-redundant sequences in there. It's about 5.3 million redundant TCR paired sequences in there, ready to go and play with. And alongside that, we have a TCR language model, paired language model, and all the things you sort of expect will go with that data. So having done that very fast, I'm now going to talk about a couple of these things in detail. And in each case, what I'm going to try and tell you is the story of, isn't this a really good idea? And then maybe it doesn't quite do what I wanted it to do. So the first of these is a language model. So Jeff very helpfully described what a language model is. We, like Jeff, ours came out in 2022, built a language model for antibodies. And ours was basically a model where you sort of mask a point in the sequence, or lots of residues in the sequence, and it will tell you what should be there. That's really useful, right? That's the kind of thing we want to do. And then we looked at this picture. So, I could have picked any antibody here. The antibody is COVOX253, and that's its actual sequence. And then sitting below that, that's its V gene. Okay? And then all we did was we masked each residue, and we asked ABLANG, tell us what residue should go there. Easy question. And we collected all the probabilities there. So you can see I've collected every probability. It's not just the top probability. Now let's take where I've got that red S, sort of the second red thing along. The S is what's in the germline. There is a mutation away from germline, a somatic hypermutation at that point in the sequence. But no, if you look at the model, it doesn't even vaguely predict that. It says what should go there is germline and has almost no probability of any other residue, and the other residues it does have probability for are not the correct somatic hypermutation. Now, this isn't a one-off example. You can look at any of the positions I've marked in red, which are the somatic hypermutations. In every case, our model goes, you know what should be there? Germline. Now, this actually isn't kind of that surprising, but we went to check. So we've got Antiberti on here, our model ABLANG, another one called Sapiens, and I put ESM2 on here, which isn't antibody specific, just for sort of a variant for you. Now, to be clear, ESM2 doesn't predict germline correctly either, so it wouldn't give you the residue you were expecting in either direction. It's the blue line. But if you look at all the others, what we've got here is the number of non-germline residues outside the CDR3. So some of these sequences have a lot of somatic hypermutations in them deliberately. And then we're looking at how often we just predict germline for all of those positions. And it's pretty much 80, 90, 100% of the time. So these models, even if you've got a highly mutated sequence, so it had 24 other somatic hypermutations in it, so you'd think, well, maybe it would notice that this position should also, no. And the reason behind that is very obvious. Pretty much every language model out there for antibodies has been trained on observed antibody space. Nice big collection of sequences, that's what we do. But hopefully you've noticed there's a broken y-axis on these graphs. About sort of 50% of the data in OAS is naive B cells. Another sort of 40%, so sort of 40% naive, 40% unsorted. And those basically have hardly any somatic hypermutations in. A very, very small number. So what that means, it's a bit like saying to a model, right, go and learn the English language. Yep, that's good. But now, what I want you to do is use those really weird words that people hardly ever use. And of course, the language model never uses those weird words because it uses the normal word for things that it's seen 1,000,000 times, not the weird word that it's seen once. And so every language model that we're training on antibody data has exactly this property. It has a tendency, and not just a little weak tendency, an incredibly strong tendency to give you germline back as your most likely prediction, and pretty much no understanding of what you would have if it wasn't germline. Now that's potentially very useful if what you want to do is say, how do I humanize a sequence? Because that's probably a good idea, that kind of tendency towards germline. Might also be good if what you want to do is stabilize it, because these are good residues to have in those positions. It's potentially problematic for a lot of the things that we think these language models are good for, like improving binding, because it seems unlikely that heading towards germline is going to be the direction for doing that kind of thing. So we asked a very simple question. Could you build an antibody language model that was actually just capable of predicting non-germline mutations? So in the same way, it would be a bit like, could I generate a model for the English language that was really, really good at speaking in Old English, words that were very rare, but it would pull them out every so often. It would do that to you, kind of play that game with your mind. This is a horrendous table. I appreciate that. But it's just to show you that doing what I do involves doing this a lot of times again and again and again with lots of different ways of thinking about it. So what we have here is a comparison of models. Right at the top there, you've got ESM2. So that has a huge amount of training data. It's not a paired sequence model because it does one chain, so you're either looking at a heavy chain or a light chain. It has a sort of standard type cross-entropy loss function, which is pretty much what everybody uses, because it works. Yep, and it can do these big batch sizes. And I've got antibody on there and ablang1. They're slightly different sizes, types of architecture they're using. And we went through a big process of trying to work out, how do I make those types of models actually get better at this? And what it turned out was you could increase the amount of data, you could make the thing paired. Didn't really make that much difference because it's just too overwhelmed with that germline information. What made the difference is you can see it changes on that loss function. At one point I go from cross-entropy to what's called focal loss. Now focal loss is a well-understood mathematical technique. It's been in the literature for some time. So this wasn't some clever idea I came up with. But of course nobody was using it because most of the time we do want our language models to kind of do what is normally done. We want a language model to use the when it should use the, and not suddenly decide, you know what, I'm going to put a weird word in there. Whereas here, what I want it to do is notice the weird stuff and learn when weird things happen, and weird things being the unusual, non-germline positions and changes. So does this work? Well, I'm going to use a measure here called perplexity. Perplexity spans from 1, which would mean you give it a sequence, you mask a position in it, and it gave you back exactly what it should have given you. Yep? And actually it goes to positive infinity. Strictly speaking, it should only go to about 20 for antibodies because it can only get it wrong, if you like, to the level of 20. Actually, spectacularly, you can do slightly worse than that. What we did to try and check if we could do this was do something nobody had ever done before, because what everybody had done is basically created the part of the table that's on the left-hand side here. Because if you just take a sequence and mask parts of it, most of what you mask is germline. So if you get germline right, you get a perplexity close to 1 and you go, my model is really good at predicting antibody sequences, it is working correctly. And you can look here, you can see ESM2 on the germline residues. It's not as good as the antibody models, but the antibody models, antibody, ablang, good at this. Right? Close to 1, you've got this. We've got good models. So what we did was we created a set of non-germline residues and said, how well are we doing there? So that's the right-hand side of the table. Now normally this number would be totally swamped, so you wouldn't see that you're getting them wrong, because if you average between the two, if only about 1 or 2% of your data is non-germline, you never see this. Now you can see that all of those models above the blue line are appalling at this. They're basically random for a somatic hypermutation, totally random. In fact, I can tell you they're not random, they're telling you it's germline really, really strongly, which means they're really, really wrong every single time. And as you can see, we started making changes to the model, and to start with, it pretty much made no difference, and in some cases made it worse. But as we introduced focal loss, we were bringing down that perplexity. So when we get to the bottom line there, ablang2, that's a paired model which is using the focal loss, which is using a very large amount of data from OAS. It's sort of pre-trained on the unpaired sequences and then trained again on the paired sequences. And we're using focal loss to try and make it concentrate on non-germline residues. Now, I still don't have a focal loss anywhere near 1. Sorry, a perplexity anywhere near 1. Now, I wouldn't actually expect that either, because we should imagine that at positions where we have somatic hypermutations, there is potential for there to be more than 1 residue. So it being perfect would be a little bit odd. And also, if it was always completely perfect, it would be useless for designing mutations, because it would give you back one answer, and that wouldn't be useful for you generating a sort of pool of possibilities. Trying to see if this is kind of really improving things, we looked at it another way. It's quite a difficult thing to work out exactly what you want to happen here. So here, on the y-axis, I've got the cumulative probability of like a non-germline change. Yep. And what I've created here to do this is sets of sequences which have the same V and J genes and the same CDR3 length, and then I found them with sets of positions where they've got a somatic hypermutation in the same place but different residues. So I can say, is it predicting one of the residues I expect it to predict in that prediction, in that place, across its probabilities? And what you can see is if I look at the non-germline, you're starting to see that ABLANG2 is actually making predictions. Its cumulative probabilities of those, they're not brilliant, but they sort of exist. What's interesting is ESM is quite good at doing that as well, because it's giving you kind of reasonable residues for those positions, and it's not constrained to germline. The problem for ESM is if I then ask it, well, can you get the germline right? It goes, nope. So it will do the same at every position. It will also give you lots and lots of changes where perhaps there should be none. So I think this is something I think is interesting. We're not there yet to make a language model that can do this really well. But we need to think the way we train these language models has set them up to do a very particular thing, which might not actually be the thing we wanted them to learn. And that's totally down to us thinking about how we use the data and what we do and how we develop with that. So my second example, I'm going to think about structure prediction. So just as Jeff was talking about, we set about building a structure predictor for antibodies. Our original homology modeling method was called ABody Builder. Yes, I do let my students name the tools. You may have heard Jeff mention Anarchy. It all gets worse. But ABody Builder, atop there, that was at the time kind of considered, that's basically state of the art for doing homology modeling. There were loads of other tools that could do that too, but we were one of them. It was like that. And then these deep learning tools started to arrive. So AbLooper was one of ours, IgFold that Jeff mentioned, Equifold, we've got AlphaFold modeler in there, and then this is ABody Builder 2 across the bottom there. And those of you who watch the literature will notice that we've literally just released ABody Builder 3 as well. I will tell you this now. It's actually not that much better at predicting structures than ABody Builder 2. I'm probably not meant to say that, but the code is an awful lot better. So if you want to get it down and play with it, I'd go for ABody Builder 3, just because it's nicer to work with. The real reason why, I show this slide, and the thing that I want to say very strongly is it is not clear from any test we have ever done that ABody Builder 2 is significantly better than AlphaFold Multimer at predicting antibodies. Okay? I could say, because pretty much every test we've ever done, it comes out slightly better, but the reality is we don't know. It's in that ballpark. It's as good. But what we like, and it's already been mentioned a bit, is AlphaFold Multimer requires about a terabyte of sequence data, and takes about, depends what you've got to run it on, but 20, 30 minutes on a GPU. What that means is, if you're DeepMind, yes, you can predict the structures of millions of things, but if you're me, you can't. And that means that I can't use it with the repertoire databases I've just been talking about. If I've got 1.5 million sequences, I need to be able to run much faster than that if I want to be able to examine the structures. ABody Builder 2 can do a structure on a GPU in about 5 seconds. Another advantage of ABody Builder 3, about 3 to 4 times faster than that. So you can do this very, very quickly indeed now. So I'm going to ask a question that sort of has an obvious answer and sort of doesn't, which is, and I'm going to use ABody Builder 2 as an example, but this would probably hold, and I have to say probably because I haven't tested hundreds of other methods, for any machine learning-based structure predictor. Can it generate novelty? Well, machine learning is a statistical problem. What we do is we give it lots of data, we say it looks like this, we train it against that data, and then we ask it, if I give you a new thing, what does it look like? And the reality of what it does is it goes, it looks like one of those, because I've seen one like it before, it's that. That's sort of what it's learning to do. So can it really extrapolate? Could it generate you an antibody structure which is nothing like you have ever seen before? And if it did, would it be right? What we're going to use to do this, because it's a well understood thing in antibodies and makes it sort of easy for us to think about it, is we're going to use the canonical form. So you have the CDRs apart from CDRH3, which have defined shapes. And actually these models are spectacularly good at predicting those accurately. So we can look at CDRL1 and it will get it right all the time, almost within, it'll be within or less than an angstrom all the time for that. And that's because there's a limited number of shapes that CDRL1 takes up, dependent on the length and sequence of that. So can it generate novelty? Can it generate, let's say, can it generate novel canonical forms? How did we do this? We took observed antibody space, we predicted 1.5 million non-redundant sequences that were in the paired set using A-body builder 2, and then we structurally classified and clustered the canonical CDRs. So what we did was we would say take CDRL1, a particular length of that, and then we'd cluster all the examples we had, and you get a picture like the one on the right there where I've got the red and the green and the blue. And basically it's saying I get 3 conformations, a red conformation, a green conformation, and a blue conformation. And then we would compare that to what we have in SABDAB, which is structurally classified and clustered the canonical CDRs, and that's been done by humans, and we use Roland Dumbrack's PyIG classify 2 as the labels. So that's what humans think there is on the left, that's what the machines think there is on the right. I'm going to show you lots of pictures like that coming up. And what we find when we do this is, and this is totally unsurprising, most predictions just map to the dominant canonical clusters. So this is the auto-clustering on the left of my massive number of structures, that's the PyIG classify on the right, and we get 3 shapes, and they map exactly, and with sequence patterns, to the shapes that you see in the canonical forms. So it's like, bang. And that's good, because that's what the model should do. It's doing exactly what you want it to do. These are things it should be able to predict. It predicts them well. The world is happy and everything is right with the world. So can we actually get something that is new? Well, these are the 3 things we got excited about, and then I'm going to explain each one is not as good as we wanted it to be. So the first one we got was we would see what we thought was a novel canonical cluster. So in this picture at the top there was no canonical form that appeared in that kind of top right circle, which I've circled, which has got the green dot in it. What was happening there was there were a very small number of structures in the data, so it hadn't been assigned a canonical form because we hadn't seen it enough times in the experimental data. But now, if we ran the sequences through it, we now had thousands of observations of it, so we now had thousands of structures, so it clustered and could become a canonical form. And actually, those were very convincing when we saw them. So it's based on a small number of structures, and clearly it's not extrapolating to a new shape. What it's doing is lots of sequences fall into that shape. And what we would get is very strong, clear sequence patterns that went with that. So once again, it's doing what we expect it to do, and it seems to be doing it quite well. The next one was a case where something would be defined as a single canonical form, and actually it was probably 2. And that's probably, once again, just to do with the volume of data. We've now got 1.5 million structures sitting in there rather than 4,000. So this example here, where that blue and green in the original was 1 canonical form, and actually you can see their shapes are quite different, because those are their shapes on the right-hand side there. So the red is one shape, blue is another shape, green is another shape. And because we've now got enough structural data, we can both see that that is a distinct shape, and we can see that it has a distinct sequence pattern. And you can see the sequence pattern on the right-hand side. So once again, that's just volume of data allowing you to better see what the canonical forms and shapes are. The last one, you could sort of argue is extrapolation here, which is we'd suddenly find, and this, say, is on the picture on the right-hand side, we would have that, and that green blob on the right-hand side was a totally new shape. There was nothing like that at that length. And we were like, yes, we're here. Well, turns out, if you just looked at the length that was either 1 above or 1 below, there was something that was exactly that shape, just had 1 more residue in it or 1 less residue in it. So what it had done was not extrapolated in the sense of made a totally brand new conformation. What it was able to do is say that these 2 things, their sequences are so similar, despite their differences in length, and the shapes of everything else around them is so similar, that I think they have the same shape. So it's like a length-independent development of canonical form. Now, we knew length independence existed in canonical forms before this. There are papers about that. We wrote one of them, but other people have as well, that showed that you could see this similarity between different lengths. But the model found that. We didn't tell it about it. It found that and generated these shapes. So I would state at this point, we have no evidence of loop conformations not seen in the training data. So you're a bit like, well, we knew that would happen. So we asked the question in a different way. Could I actually just force it to do this? Let's make it kind of like, come on. So This is that same kind of plot, but I'm just showing you kind of the test set of sequences here, so there's far fewer dots on it. So I have SABDAB on the left, so that's the actual structures, that's the actual shapes of these things. Then I have how I normally trained A-body builder 2 and its results in the middle there. So look in the top left here, this is a CDRL1, length 13, and you can see A-body builder 2 predicts those purple things to pretty much be where those purple things should be. So good. What I'm now going to do is I'm going to retrain A-body builder 2, and that's what's called the starved model there, and I'm going to delete every piece of information we have of anything that is in the top quadrant of that. So it knows nothing about that shape. It's never seen a shape like that. And this is kind of the interesting result, I guess. So those purple structures now sit in a slightly different place. They're still in that top quadrant, top left quadrant, but they're definitely not the same shape anymore. And we've done a similar thing across the bottom. You can see a very similar thing. So SABDAB, we've taken the green ones out. A-body builder 2 predicts where they should be because it had data about those. We completely starve the model. They're not there. They now suddenly appear in the top left quadrant. But they're not appearing on top of something else. So what's actually happening here is, yes, it can predict a novel conformation, and we did this lots of times. I'm just showing you 2 pictures. It does predict a novel conformation because it predicts something it's never seen before. Unfortunately, it doesn't predict the correct novel conformation. So what happens, I think, in these models, and it's an interesting kind of statement, is when it predicts a conformation that has never been observed before, it is right that this thing does not have the same conformation as anything it has seen, but it doesn't know what it does have. It just knows, it's not like any of these, so I'm going to put it there. But it doesn't know where there should be. So the final test was, well, how much do I have to tell it till it becomes good at this? Do I have to have tons of data? Because if I have to have tons of data to make this work, there's sort of no point. I'm going to lose this war constantly. I might as well just let the experimentalists go on with it, because I'm always going to need them to solve something before I am useful again to be able to tell them something. It turns out you need almost no data. So you've got a model which has a base large amount of data, and we do this starved trick again. So in this picture, SABDAB's on the far right, and then I'm showing you how badly the model is doing. So A-body builder 2, it's results which have like the data inside it should be able to predict things in that quadrant. So if you have no data on the left-hand side, it doesn't get it right. So those blue data points, which should mostly all be down in the bottom right, it sort of splurged them all over the place. I add a very tiny bit of data back in, and it's like you're talking 5, 6 structures. All of a sudden, bang, it starts to know where they should go. And as I add more data, it gets better and better. We found that less than 1%, so tiny flicks in the data, allow you to make this step through to actually having a useful predictive model for something. So that suggests that targeted solution of things and having very small amounts of data about different structural shapes, these models are very good at picking up that that is a shape I haven't seen before, but all these sequences belong with that shape. And that's, for someone like me, that's really good news, because all right, I can't do everything brilliantly, but every time the data changes even a tiny bit, my models should improve and should get better at doing the things we want them to do. No? Forwards? There we go. So I'm going to finish leaving this up for a couple of seconds to make sure everybody just appreciates all the massive amounts of hard work that my research group actually does, and then properly finish by saying thank you to all of them. So on the right-hand side, that is most of my group when we went on a group retreat, and on the left-hand side, we had a reunion a bit over a year ago now, because a lot of what I talk about depends on the work of people who've been doing it for a very long time. Any of you who've been involved in data curation will know that the effort of making OAS or SABDAB or OTS is not the effort of one human, it is the effort of a group, continuously having to do that over many, many years. And I will finish, and I always say this, and I'm never sure people quite believe me. I'm going to finish by saying thank you to all the people across the bottom there, those organisations, because they literally give me money to do the job I really enjoy doing. Thank you very much.
Well, thank you very much, Charlotte. That was so inspiring. We have a ton of questions for you, if I can ask you to try and be as concise as possible with each of these. First one is from Professor Jeff Gray, asking how are OAS and the AIR data commons related? Is one a subset of the other? Are the underlying data sets the same?
I think I should let AIR answer this better than me. But they are related in that OAS operates to the standards of the AIR commons so that they can be integrated in. We've made a very specific choice that ours are full-length antibody sequences and full-length paired data, and with TCRs it's the full-length paired data. And then we have some annotations on them that are kind of slightly different, but they are integrated together in the sense that all can be within. Yeah.
Perfect, thank you. In reference to your language models, could the skew towards germlines be caused by your temperature setting?
So the difference there is that, yes, you can turn up the temperature, but what I was showing, trying to show the probability graph, is that what that will do, yes, it will make the ones that are below more probable, but the issue with that is, well, it will make you more likely to choose from them, but unless you change the loss function, those less probable ones, so that little bit at the bottom, so it was germline, tiny bit down here, those are not even vaguely in the right order. They've got nothing to do with somatic hypermutation at all. The model doesn't have a clue what it should be apart from germline, basically, unless you try and tell it that it has to know what it should be apart from germline. Yeah.
Granted. Regarding your perplexity numbers, again, why is it possible to predict the CDR3 contents to any extent? Isn't it supposed to be random?
So CDR3 is not random. I mean, CDRL3 is certainly not random because it's encoded very precisely. The beginnings and ends of CDRH3 are also not random. There are also structural, because they come in from the parts of the gene. You also have to remember, I mean, Jeff showed it a bit, that in a particular structure, you definitely have preferences for lengths of H3. There are definitely preferences for sequence that will actually create a shape. So one of the things you can test is how well, if you're building language models and generating sequences, I mean, Jeff showed this, you can look at how well it will make a structure. It's very interesting to look at how well an H3 will actually generate structures that make sense in those models. So they're not random. They really aren't, but they have just a very, very large number of choices. And that is dependent on everything else that's around them. So we showed in a paper that the whole kind of sequence and structure space of the H3 does depend on the other CDRs that are around it, so the rest of the framework and everything else.
What are the use cases for antibody structure prediction not in complex with antigen? Or is this more of a step towards doing antibody-antigen complex prediction?
So some of them are, I mean, there's an awful lot where it is directly, yes, I just want to predict the structure of the complex. Some of it is also for us to be able to do other things, like, for example, I didn't go through them, but if you want to predict the developability of your antibody, it is way better to be doing that on a structure than it is to be doing it on a sequence, because you want to know what's on the surface of the antibody, you want to know how that would interact with other parts. And many, many of the properties we'd like to predict are predicted much better if you can have the structure rather than just the sequence. So that's one thing. And another thing is it's very useful to be able to predict at that rate, because then you can use it as input to all the other algorithms you want to build, so that then you can build a better version of, I mean, Jeff gave one example of kind of putting in AlphaFold 2 structures in to kind of improve your predictive ability of just antibody structures themselves. But you can also do it for the docking thing. I'm trying to go fast. Working.
Thank you. One final question here from Brian Corey. Related to Jeff's question, how can we combine the data in multiple repositories, like OAS and ADC, to better train these models? For example, do we need more types of B cells?
I think, I mean, one of the issues with combining the data is always that actually most of us are collecting the data from the same places. So actually you don't gain much by putting lots of the databases together, because what's happened is we've just combined, we collected from the same papers and things, and we're just trying to clean it in different ways. The other thing to always worry about slightly when combining data, and this is particularly prevalent in antibody sequence data, is everybody has a particular pipeline they use from the I got this off the machine to here are your amino acid sequences. If you run the data through two different pipelines, you get two different answers. Now, they're not massively different, but they are different enough that it can make it appear that there are statistically significant differences between data sets when there are not, because of the scale of these data sets. So, for me, it is about ensuring that we have places where we know that everything has been consistently processed, and then you can work out how you can work on forwards with them. But it is a continuous challenge to get as much data together as possible for this.
Outstanding. Thanks again, Charlotte.